So thank you all very much for coming, and thank you, Mike, especially for inviting me here, being so kind. It's really nice. I was very happy that you kind of saw my talk and apparently liked it. Yes, I'm currently, as Mike said, at the Center for the Future Mind, Florida Atlantic University, but unfortunately, due to COVID reasons, I have to stay at my home in Europe at the moment. Well, so that's the current situation. I hope it gets a bit better here. And what I will talk about today, the title of my talk is kind of Constructing Spaces. And I'm particularly happy to be here because my initial background, my PhD was actually in physical chemistry. And part of my PhD was the question whether one could use molecular setups to actually construct or build interesting things. We're working a bit on molecular switches. That's a very primitive thing, but one can imagine that in more complex molecular systems, biological systems, maybe one can really get to cool things. And that's basically what you are very famous for, kind of constructing cool things out of molecular matter, biological matter. Over the last years, after my PhD in physical chemistry, I started philosophy. And there I was very interested in these big ideas and concepts and ways how we describe the structures of our thinking. And there's an interesting question how one could relate those two things. So the molecular biological side on one hand, and the philosophical conceptual side on the other hand. And that's kind of interesting. That's an interesting question. I had the pleasure to spend several years prior to my engagement in Florida at UC Irvine in the lab of Don Hoffman. And he had this idea of interfaces, interface theory of perception, which kind of brought down a lot of these big philosophical concepts to a very let's say, operational, tractable form. And of course, the interesting speculative question in the end is, well, if one could use molecular stuff to realize certain high-level concepts, but those concepts are something like an interface. I will talk later about it, what I mean with this. What does it actually imply for the molecular stuff one was working initially with? So I start by introducing a little bit some philosophers. So excuse me, probably you've all seen that quote. It's by Galileo Galilei, and he made this distinction between mental properties in consciousness and material objective properties like space, geometries, shapes, and on the other hand, the subjective properties like tastes, odors, colors, and so on. And he said, I think that taste, odors, colors, and so on reside in consciousness. Hence, if the living creature were removed, all these qualities would be wiped away and annihilated. Galilei's distinction made a little career later on. John Locke, a British philosopher, introduced the distinction in primary and secondary qualities. So a lot of people, and I think a lot of people still have this intuition that there is a difference between some are more fundamental objective properties like space, and properties which are kind of less fundamental, which are subjective, which do not have the same status, like taste, odors, colors, and so on. Now, a couple years later, there was another philosopher, Immanuel Kant, very famous German philosopher, who kind of revisited this idea of this distinction into primary and secondary properties. And he said something like, now, if I go farther and for weighty reasons rank as mere appearances, properties such as extension, place, and in general space, no one in the least can adduce the reason for its being inadmissible. So what he means by that, he says, well, let's think for a moment. When we talk about space, extension, place, shapes, geometries, maybe those kind of properties have a very similar status as the so-called subjective properties that Galileo mentioned. A third philosopher, which I kind of like in this context, is Alfred North Whitehead. It's not so well known as the other ones, It was roughly 100 years ago. And he kind of tried to argue for a middle way between the materialism of Galilei and the idealism of Kant. He was advocating a concept of nature, which kind of included both subjective properties as well as objective properties. And he said that it is for natural philosophy and for science to analyze how these various elements of nature are connected. And I'm mentioning Whitehead because he had a very interesting idea. He later wrote a book which is very difficult to read and like a big volume and very involved, called Process and Reality. It's his big metaphysical work which he wrote at the end of his life. And another name which is often associated to that project is the philosophy of organism. So Whitehead believed that in order to get such a concept of nature across, a special metaphysical idea is actually needed. And he introduced the idea that nature or the world more generally consists of these little, he called it actual entities, which were kind of the atoms of his systems. And the interesting thing in this context is that if one looks at what he says about how one should think about these entities, they are very biology-like. So they have intentions, somehow goals, and on a very fundamental status. So Whitehead believed that one could or one should regard those things as fundamental and try to look whether one could abstract from that typical properties such as space. So that brings me to my to the actual topic of the talk today. I mentioned Don Hoffman, whom I had the pleasure to work with. He had a somewhat similar idea. He said that our perceptions evolve to keep us alive and not report the truth. And the way how Don Hoffman is putting things in his book, in his recent book, space-time is just a data format, much like data structures in your mobile devices that serves to keep us alive. Our senses report fitness, and an error in this report could ruin your life. So I think a very strong and catchy statement. He also introduced a metaphor, and that's the interface or the desktop metaphor. Perception can be compared to an interface that hides the complexity of our environment, and space is our species-specific immersive 3D desktop. Real-world objects are mere icons on this desktop that represent fitness consequences. So I think that's a very interesting and intriguing statement. The question is, how can one make those statements concrete, and what to do with this species-specific immersive 3D desktops. Now, another famous mathematician, Henri Poincaré, had in some sense, a very similar idea. He was thinking about a particular problem, namely the problem of how to get a notion of space based on sensations without having a spatial reference at hand. So if you have a spatial reference, then you know a certain sensation that you have is associated to a certain point, and then you can construct the space from there. So that's kind of trivial, but Poincaré had this thought, oh, well, if you don't have this, and that's probably the situation that we are all in or that organisms are in, what to do then? And then Poincaré had this nice idea that there are actions which could compensate for external motions. So when you're situated in some environment and there's something moving, you could move in an opposite direction, and that would leave your sensations invariant. So that was his insight. And then he derived from that one could perceive the sensory consequences of these actions, the compensatory actions, and from those sensory consequences construct the concept of space. And that's exactly what I was thinking about recently. Now, first, maybe what do I mean when I say space? That's a very big notion. one can, I think, break it down to a mathematical notion, which expresses or which refers to some symmetry. So basically, when I say space, what I mean is a certain set of points, which is structure, which can be described with the action of a mathematical group. And an example would be, for example, would be Euclidean space. And there's a Euclidean group which describes translations, rotations, and reflections on this real space. So that gives you this idea of a classical Euclidean space. An important point to think about is that symmetries express invariance. So the normal way how we recognize symmetries, we see invariance. We see that thing which stays the same under changes, change without change. That's a kind of catchy definition for symmetry. And if we go back to our example, to the Euclidean space, if one applies the Euclidean group, that leaves invariant certain properties of objects, in particular the distance between 2 points, which is the norm on R3. A third point in this context is that such spaces, they need not kind of exist outside, or can be inferred from what there is outside, but they could also be invented by an organism. They do not need to reflect the property of an object, but they could reflect the property of the way how we interact with objects. So there's this kind of enactive component, which I find very interesting. Okay. So I was thinking about a way how to kind of implement those in a very simple toy model-like setting. And I called it a kind of symmetry making machine. So what you need for this way of constructing spaces is basically a set of sensors which record certain information about the environment, about, I will talk about light sources later on, but could be anything actually, and which also need to record some feedback from actions which an organism can take. Then, of course, an organism, or the symmetry making machine, which how I call it, needs to be able to make some actions which acts on the environment, which has some consequences. And in more evolved setting, there will be a certain mechanism which connects those two things. But initially, the first step, which I will talk about, is if there's just a randomized relation between those two things. So there is no sense of control at the initial step. One could also think about there's a kind of learning stage where one just doesn't engage in any sophisticated controlling behavior. Now, a question for, I would say, practicing scientists is, of course, how and whether one could implement such a machine, and then what it is good for, what one could do with it. So I'm more focusing on the abstract description of how these things work, and I worked out a little toy model over the last weeks, which is a toy model for such a little embodied machine.
Sorry, can I interject and ask a quick question? If that's
okay? Sure. Every time.
Yeah, it's on the previous slide. You call it a symmetry making machine. Could you explain a bit more where that symmetry is? Is it the isomorphism between A and S, or is it something else?
No, the symmetry will be a symmetry between past and future recordings of S, which is mediated by actions of A.
I see.
So maybe when I go to the toy model, it will become a bit more clear. So the idea is symmetry is very much related to this concept of invariant. So if you record something which stays the same, then one possibility at least is that it's due to some symmetry.
Yeah. Okay. All right.
So if you look at this minimal toy model which I produced, there's just a small, well, agent, maybe it's even a big word. There's like a little retina on this grid, which can move freely around on the grid, and which can also record certain sensations, in that case, light intensities, which are coming from various sources, which are dispersed. And what the machine will do, it will record at each time step, it will record light intensities. And from this recording, it will try to infer a space, in a sense that from these recordings, it looks for invariant, one can look for invariants, and those invariants might be used to infer the concept of space or construct the concept of space. That's what I meant with symmetry making. Making is a bit strong. One can argue about whether it's making or whether it's recognizing, but that's a different point. And the setup which I used later on, I have 4 proprioceptive sensors, but the number is arbitrary, so you can do with different numbers, which just correspond to the distance from the corners of the grid. But as I said, it's arbitrary. The more you have, the easier it is, obviously. So it works with much less. But the good thing, the more information a system gets, the easier it is to kind of infer the spatial concept. And the easier it is, you will see that in the next slide, the less ambiguity it can have. So sometimes it gets signals which are, so it's in a situation where it has similarly whereas various candidates for invariance, and it needs to select one. And the more proprioceptive and sensory signals you can compare to each other, the easier it is to make this selection. So a concrete model, before I come to the inference part, one could think, okay, we have this agent which can move around on the grid. One can present it with a collection of light sources. Then one introduces displacements, and then one just see, okay, what's the difference between before and after? Here on this slide, that's a very simple example. It's just a number of light sources which are dispersed around a circle, and out of the difference data, it can calculate this displacements, at least on average. So that's a very, very simple way to think about it. I also intentionally kind of chose a very simple geometry, a circle. So in principle, what I will do afterwards works for various geometries. That's a nice thing, and actually randomized it over my little computer simulations. So, but there is a problem to this very simple picture which I just presented. That it's all good, but if you recall the problem that Poincaré kind of raised, well, what happens if the agent doesn't have any access to some spatial reference? So if you compute like differences, for example, then the way how you usually do it, you would say, okay, take the position here and subtract the position here. That's obviously not possible if you do not have any reference point. So what should you do in that situation? Well, the machine knows the sensory consequences of its actions. So what will happen to your sensory input after movement? And the idea is, what I just said before, that out of the sensory inputs before and after movement, you can construct equivalence classes, for example, invariances, which then encode something like the symmetry of the spatial field of that organism. So more technically spoken, the equivalence classes induce a certain structures on the set of proprioceptions, which could be used to infer space-like representation. For example, the easiest thing would be just classifying those displacements. And
can I ask another question here?
Yeah, sure.
So for the agent even to make those actions, right? For example, to move, do you think it would also need some sort of spatial reference? Because I don't know how else it would move without
Well, spatial reference well, it depends a bit on what you exactly mean by that. So I don't see how it needs to be able to say, well, move to this particular position and give this particular position a certain number in some coordinate system. There needs to be some sort of movement, and of course that happens in space. But I don't think it needs to have a spatial reference in the sense that you're able to kind of say, oh, move to the place X in the coordinate system here.
Can I also ask a question here, Robert?
Sure.
It seems to me that having the proprioceptive sensors actually constitutes a spatial reference frame, in that the organism is able to detect something that correlates with its motion. I mean, contrast this agent with an agent that's exactly the same but doesn't have proprioception. It can still move around just fine. To answer Santosh's question, it just doesn't know that it's moved. It has no way of knowing that it's changed its position, even though it has, in fact, from our point of view, changed its position. Whereas when you add the proprioceptive sensors, there's something that correlates with motion, and I mean the organism may not be able to remember that correlation, but the way you set it up here, it does. So I think you've built in a spatial reference for it.
Yes, well, in some sense, I mean, I definitely would agree that you have that you know about something which correlates to those motions. But still, from the correlates, you need to do something with the correlates, I guess. I mean, if you just have the correlates, then it's interesting, but it's a bit of a complicated mess. The question is how you can extract out some specific notion of a displacement, for example, from these correlates.
I mean, yeah, you certainly need a comparison function in a memory.
Yes.
To turn it into.
Of course, you need, I mean, you're perfectly right that you need a memory and that you need to make this assumption that one could, or that's an interesting question, how that could be realized, that one could record instances or sensations or recordings of movement or whatever. I think that's a precondition, definitely, yes.
I mean, I'm visualizing, for example, a biological system that is basically firing off various signals, but from the inner layers, you don't even know, right? If you, for example, on the effector side, if you fire off certain signals that may or may not connect to muscles, and you may end up moving as a result of it, but the system that's generating it doesn't need to know, or it often doesn't know what's going to happen, right? That's even a movement. And I think the same thing on the sensor side is, you know, you're eventually, what you're really getting internally is maybe some calcium spiking or something, and you don't have any idea where that came from. I don't think there's necessarily a spatial a spatial component baked in, because all you're receiving are these different channels of stimuli and effector activity that may or may not result in moving.
Actually, that's an interesting point, Mike, if I may add to it. But if I could be the devil's advocate for a moment, all of these organisms have evolved in space, right? So is it possible that all of this calcium activity, neural activity, have fine-tuned or calibrated to the space they have evolved in? Do you think that's possible?
I actually think that if you think about the evolution of the simplest life forms, I think the space that we think of as space, as, you know, three-dimensional space, is actually one of the later spaces that life learns to handle. So I'm immediately thinking of metabolic space, you know, transcriptional space, these kinds of, you have to take actions, and I think this whole formalism maps very nicely onto these other spaces where, in fact, you're not moving in the way that we think of as moving. You're changing your state in these other quite what we would think of as virtual, but to the system equally appropriate spaces that have nothing to do with translocation in three-dimensional space. And there again, you have these, you can take actions, meaning you can, let's take transcriptional space. You can take actions, so you can turn genes on and off. You have proprioception in the sense that you can tell when certain genes come on or certain proteins get tickled. And there are better and worse regions of that space for you. And to have a successful adaptive life, you need to navigate that space. And you can, and creatures that don't move at all have to do all that kind of stuff. So all the way from, from let's say, simple plants or single cell organisms that are stuck in place. You don't have to move. There's a ton of other spaces, not to mention morphospace for metazoans and so on.
Yeah, the plant example is interesting.
Yeah, I think an interesting aspect of that comment, Mike, is that in biochemistry, there's an automatic memory, which is just the lifetime of concentration, right? Molecules don't decay immediately. So there's an automatically available and sensible memory for how much transcription, for example, has been performed, because you have the concentration of that molecule, which has some effect.
That's right. So you get, so in effect, then you would get in that space anyway, you get these sort of comet trails, right, of directionality, you get that.
Right.
Yeah.
I mean, the interesting question is how you can access, so to say, the, how can you, in a lab, you can measure the lifetime in some sense. But the question is how a simple organism kind of goes on about this. I guess, I think probably it's straightforward and possible, but it's kind of interesting to specifically look at the implementation of that. So I'm thinking about similar to a chemotactic-like mechanism, which is basically very similar. You have a lifetime of your phosphorylated stuff, and that goes down, and that changes, at least probabilistically, the behavior of the thing. I don't know, just my guess. So in general, I think Mike thinks for your comment, for your clarification, I think that's a very good comment, actually. So when I said on a few slides earlier, the notion of space which I have in mind is really this kind of very abstract notion of a set of points and a structure. That's more, that's it, and the I would say the illustration with things moving around in space, in physical space, that can be kind of misleading, because you automatically come up with this question, but well, typical questions, and in the end you always say, oh, but then the best thing which you can get is basically that you infer the physical space which is already there. But if you really think about different kinds of spaces, that intuition kind of goes out of the window. Okay, so just an exemplary workflow. What I did in little simulations, I took a certain number of random displacements. So in the interval 0, 1, but randomly, so all around the unit circle. I created reference geometries, also randomized random dots within a circle, applied these displacements, and then I recorded so-called scans, where the retina is able to move around the grid, and you can just store information about sensory values. And then in the next step, it does what Chris was talking about when he mentioned the correlations. It can look about how things, or how sensations correlate to other sensations across different time points. And out of that, given a certain criteria, for example, delta SE 0, but one could also use different ones, select certain, or limit this huge data set down to a smaller set, which could then be used to infer something interesting. And as every time when you do not really know what to do best, you can think about a neural network. I just fed that into a neural network and tried to learn those displacements. And the thing is, it really works kind of nicely. I know that numbers here are quite large. Well, not very large, but little large. I tried it even with smaller numbers. And what I found was actually a very nice convergent behavior. And those displacements could be guessed for training sets with less than 5% error. So it was I was kind of happy about that. Of course, in a real biological system, for example, you wouldn't think that you have a neural network available to do this kind of thing. So you would need to think about ways to actually do those correlational analyses or those calculations without this kind of involved technology. So main question which I had is, which we just talked about, whether this could not only be applied to physical space, but to different sorts of spaces more generally. In principle, I think it should work, as long as you think that space is just a very highly regular way of representing potential and actual inputs. In particular, and also if you get feedback about the ways of behaving in that particular space, but it doesn't even need to be intentional behavior, it can be just randomized behavior. The big question is, well, if you do that and if you construct such a spatial representation, what is it good for? One immediate thought which I had, there's a kind of truism in computer science which says garbage in, garbage out. So every empirical scientist probably knows if you record some data, if you do an experiment, for example, there's a vast stage of pre-processing. So usually you have to massage your data in order to do something useful with them. And one function of space could be just something like such a kind of pre-processing of raw sensory data. Then there's this nice idea, which actually Chris had, that space, or which Chris wrote a paper about, that space might be an error correcting code. I think that's a very interesting idea. I won't talk about that any further. But those are two kind of possibilities for how spaces can be very useful. Going back to the implementation question, as I said, my background is in physical chemistry, so I'm kind of always interested in this question. Oh, well, think about a easy, simple molecular system. How could you think about these little machines? And you're obviously the experts on these kind of questions. Well, so I don't need to say not need to say much about this. I think it's obvious. Sensory systems could be receptor systems, which are coupled to kind of molecular machines, molecular motors. And what you would want to have in the end is a kind of like a chemical computer, which is programmable in the end to make this inference about correlational structure. The nice thing, because it's so simple, it's kind of cheap, you can have a lot of them. One could think about parallelization, so not only invention of space within a single organism, but something like a shared space in a collection of organisms. And the third point that connects back to the interface idea, that these spatial representations could serve as a very interesting interface in the sense that it enables organisms or these machines to have to choose certain adaptive actions after they learned this concept of space. Well, there are different ways how one could think about how one could implement it without appealing to like a neural network style architecture, which I had before, a simple reinforcement paradigm with an evolutionary algorithm. That's one possibility. Perceptual control theory, is another possibility which I found intriguing. I just recently read about it a little bit. The idea with perceptual control theory is that you use a hierarchical system in order to modulate those criteria when, for example, when you find an invariance or not. So I had the criteria of delta S being 0 or close to 0. One could think about an hierarchical system that actually modulates those criteria. Well, okay, so I think, yes, I think that was it already. So maybe we have some nice discussion. If you have any question, I would look forward to it. Thank you.
Great, thank you. Very interesting. I've got a quick question. What do you think are the, to me, one of the most the interesting things about this that I think we can use is to really extend this to the notion of these other spaces that life solves problems in. So the things we worry about, they solve problems in transcriptional space, in metabolic spaces, in amorphous space, anatomical space, and so on. And so I'm interested in these symmetries and how they can construct and solve problems in these spaces. What do you think are the minimal necessary components for an agent that is able to do this? In arbitrary spaces, like what's the core thing you need to have to be able to do this?
Well, I think the most important thing is that you have a feedback mechanism. I mean, you get some feedback for whatever you do. And of course, it works well if that feedback is quite quick. So I mean, if you have to wait for a very long time, then it's different, at least on the individual level, to do this inference. So I think that's a kind of that's probably a minimal requirement to make it work. In principle, I think. The more that I said it in my talk, the more ways of feedback you can get in some sense, the better, because that might give you more structure or more correlations to actually work with. I don't know which particular spaces here are suggestive for that. Other than that, I think there's no requirement, but of course there's this question, even if you could do that, what would you use it for? And what's the benefit of having this? I mean, it's something like at least conceptually, it's a bit of an involved construction. So the question is how could you benefit from it over and above, for example, just exploring randomly your environment.
What kind of feedback are you talking about? So I can see at least two kind of feedback. One is the in-self feedback, meaning the organism know exactly what he did and can compare the environment after its action, or it doesn't remember what is done, and just sampling the environment that it's intervening on that environment. In that way, the feedback is one step further, but it's delayed some sort of, but it's still a feedback.
I see. Well, I mean, that's a question of memory in principle. I mean, whether you're able to how to record, and that really depends on the way how you set up the algorithm. I mean, in this toy example which I had with the neural network, of course they assume you have a lot of memory, but in a more realistic scenario, in a biological scenario, for example, you probably won't have this immediate feature that you can have as much memory as you want. But I mean, I think that's a question for the particular algorithm which you might want to use. If you just think about calculating and storing differences, then it's probably, I mean, I guess at least it's probably something which you could implement quite straightforwardly in a chemical system, for example, I guess. But still, I mean, if you need to have, if you want to have memories over many kinds of movements, then it might become a bit tricky.
Right, but in this scenario, that will be also considered to be a feedback in that system.
Oh well, I think when I say feedback, I don't mean that I don't imply that it could be stored endlessly, for example, or that you have infinite memory capacities. It just needs to be some response immediately after, or maybe not immediately after, but after the sufficiently short after the action which you took, because otherwise it's difficult to kind of make these correlations, I guess.
What I really like about this idea of nullifying changes in the experience is that, to me, that sounds exactly like homeostasis. So when you have a basic homeostatic loop and you're deviated out of your comfort zone, what you're trying to do is figure out, okay, what can I do to nullify this and get back to where I was? Which action of mine can counteract whatever is happening? So if that's hunger, or if that's injury, or if that's whatever is happening to you, it seems like all of homeostasis is learning to nullify whatever is happening so that you can get back into your optimal range.
Yes, I think that's a great way of thinking about it.
So it seems to me that you can build this up from scratch, from a very minimal unit that all it knows how to do is try to homeostatically, sort of keep alive, basically. And the next level of that is this kind of more complex scenario where you're actually now remembering the steps that you took to nullify these various things, and now you're starting to build this up into a space. Like, that sounds like a construction of a basal cognitive agent to me from the beginning.
Yes, I think that's also kind of, I mean, at least that's how I interpret him. I mean, I talked about Whitehead in the beginning. The philosophy of organism is probably very close to the way how he was thinking about things. I think, coming from with a physical perspective, it's kind of difficult to wrap one's head around what he actually thought and said. But probably if you look at it from a more biological perspective, or if you limit it to kind of biological systems, in some sense, then it makes much more sense, or it's much easier to digest.
Going back to this discussion on different kinds of spaces, right? Physical space, martial space, genomic space. So one thing that I think it goes back to Robert's premise that spaces is a kind of invariance, right? And so when you ask what is a necessary condition, perhaps one is that if you consider genomic space or biophysical, biochemical space, there must be some kind of invariance that organisms must be able to pick up. So that's one. And the other is there must be some way of constructing sensors and actuators that can effectively pick up that invariance. So I'm just thinking out loud here, and it might not make sense. For example, when it comes to genomic space, there are these genes which are, in a sense, invariant, in the sense that they are quite stable, and it's possible to read off information off of it. And perhaps the elements and molecules or whatever is available on Earth has made that possible. And perhaps that's one reason why life cannot evolve on a more chaotic environment like Jupiter. And also, perhaps life cannot evolve in a place near a black hole because space itself is not invariant. It can be destroyed. If that makes sense. I hope I'm not rambling here.
I mean, the invariant that I immediately thought of when we started talking about this is that we had this the study of planaria, these flatworms, where you throw them into a general potassium channel blocker and their heads explode, and the cells can't take it. But after a while, they regenerate these new heads that are completely barium adapted. They're fine. And we looked, and there's basically a small number of genes that were upregulated to enable different channels to enable these things to do their business without using all the stuff that's blocked by barium. So that to me is an invariant, right? It found a move in transcriptional space that allowed it to do all of the stuff that it used to do. So from the perspective of the physiology, nothing has changed. But from the perspective of the transcriptional space, you had to make a move to counteract what happened, right? So that type of invariant, that it used to be this set of genes, now it's a slightly different set of genes, but what's invariant is that with both sets of genes, you can manage to do the basic, physiology that the cells need to do.
Yeah, so for me, when I think about invariance, it's really about keeping invariance the relations between actions and sensations, and not the invariance of some structure out there which already exists. I think that's conceptually the thing which I would associate with this invariance.
Hey, Robert, I had a question which kind of centers around the idea that if you have a bunch of different spaces that are, conjointly being explored and being utilized, at what point do you find an optimal number of space that you actually actively try to sample? So, for example, right, we all know this. You might know this example where, if two people try to find each other, it's always more, it's a better strategy of one staying in the same place and the other doing the exploration. Otherwise, you have too many combinations of where they could lose themselves. And I was doing these simulations based on Bayesian inference, where you had essentially two spaces that you were exploring. One was just like a cell signaling concentrations, and the other one was movement. So these cells move around in a space, and they could basically sample the concentration of these signaling molecules at each position, and they would try to basically find each other and find out their own identity based on those concentrations. And when I tried to actually get rid of the movement, thinking this may actually be easier for them to just basically stay in the same position and just they secrete signalings and then they get more signaling concentrations back. So they should be able to do this just by sitting on their own position. And I had some technical difficulties, but it made me think of at what point, can you kind of speculate on what would be an optimal number of spaces that you would want to explore simultaneously, like transcriptional, actual moving space, based on I'm guessing the answer can be based on the time scale that those things interact with each other. But I don't know if you have an idea based on your modeling.
That's a very difficult question. I have no intuition about that.
Have you tried modeling your toy model on two different spaces simultaneously?
No, actually, I haven't tried that. I think that's a very intriguing idea. So I was thinking about how you can combine various modalities within one space. But the more interesting question might be actually to have different spaces for different modalities and then look at cross sections here. But actually, that hadn't crossed my mind before. So I don't have a good answer, but I think it's a very interesting.
Cool.
Yeah, I mean, we should definitely talk about it, because I think that one example, we have other examples, but that barium thing is one example where you have two spaces that are linked, right? You have this physiological space and you have this transcriptional space. And we also have other scenarios where, for example, the genetic space, maybe that Santosh was talking about, with the exact same genome, you could make frog embryos, or you could make xenobots, or you could make various other things. And so in anatomical morphospace, there are differences, but they all map to a kind of invariant in genetic space, because they all have the same genome. So there are lots of interesting examples of the multiple spaces being linked in that way.
I think that Franz made a really important comment in saying that different spaces come equipped with different time constants almost automatically. So the genome space has a much longer time constant than the transcriptional space, which has a longer time constant typically than the movement space. So one can sample spaces with different time constants in a kind of hierarchical way. And if you think about disrupting that hierarchy, then that screws things up, or it can introduce real advantages. For example, lateral gene transfer changes the genomic space, maybe very radically and maybe very quickly. Of course, it can be lethal too.
Yeah. I guess what I'm specifically wondering if those spaces are actually, do they have to be linked? You can make a null hypothesis saying that, oh, well, the cell has just like all these different spaces to explore that come with different time constants. So it's going to try all of them. And, if the faster one works, then great, but the faster one might just not have the right precision, not the right interaction with the actual environmental signals. So like at some time scale, it's gonna work, but I doubt that's true. So I wonder what the actual implementation of a linking of those different spaces in time and space would actually be. And like, so like with my simulations, what I was thinking is actually that, there's something fundamental about when you move in space. It gives you a lot better, I guess, essentially 2 more degrees of freedom of creating a gradient descent, right? If you're just sitting in the same position and you already have all these signal concentrations, you move them up and down, but it's hard to I could imagine it being hard to actually make an inference of what that really means, because there's too many combinatory factors that could, overlay. But if you then can sample them in space, you get a much higher precision on a stronger causal relationship there. And that's what I'm wondering, if those spaces, how much would an organism or an agent, how much would it utilize the interaction between different time-scaled spaces to make inference about its environment, as opposed to just having different mechanisms that just kind of run in parallel, just in different timescales.
I think that the organism, if the organism know that there is more than, like, if you give them oracle clue and you're going to say there is 2 different distribution here, it will be much easier for the organism to get know the parameter space, or to understand the parameter space. But the real question, it will be how the organism know how many kind of different distribution there is in the space.
Mm-hmm.
Well, one could imagine an experiment where one does something like construct a situation where E. coli are in a plate, and they have a glucose gradient and a fructose gradient or a galactose gradient that intersect. And you can see how many of them try to solve the optimization problem by moving, and how many of them try to solve the optimization problem by changing their transcriptional state and hence their transporter array.
That's a great
Right. But is that because there is different kind of mechanism that each one of those mechanisms specialize in the fructose or glucose or something, and the other mechanism cannot handle the other chemical? So it's only one mechanism per chemical, and then what you have is a battle between those mechanisms, to which one of them are dominated, or where the organism as a general will go, because you have 3 different directions.
Yeah, I think it would be a competition between, you could solve the problem by moving. And so if you happen to be set for metabolizing glucose, which is what they like, then they can maximize that by moving. Or,
right,
or they can say, well, I'm not going to move. I'm just going to readjust my metabolism to deal with galactose and stay put.
But that's means saying that the organism know that there is 3 different concentration. So there is no 4th, or is not aware of the 4th.
And I wonder if it's really true that the organism can choose, not taking all the baggage of that, but like fundamentally in like an inference scheme, an active inference, if you define a certain like generative model as some kind of goal state, right? Those 2 things are always linked. Like an organism, like at least in my simulations, and I think it's true for all of these generative models, that essentially you define what kind of position they want to have certain signalings at. That's the kind of their target morphology essentially. And if they are not happy where they are, if the concentration of the sensing doesn't match what their position belief, they will update both of these parameters. It's because they only have one map of position and signaling concentrations that are linked into one target. They're not really capable. That's why my simulations failed when I try to separate it, on a technical level, but also fundamentally the way it's set up, it's one target, right? It doesn't view those things as completely different ideas, and I wonder if that is just a quirk of active inference schemes or my solutions in particular, or if that's something more fundamental of this idea that if you define a certain goal state based on a certain number of spaces, then those are going to be linked no matter what the timescales are. And you just basically design different strategies. The strategies are only separated in their efficacy over time. So like essentially they're doing the same things, but because some things work faster and something works smaller, it's going to appear as if they chose one over the other, one strategy over the other, one mechanism over the other. But in reality, those are all linked together with one fundamental underlying goal structure. I don't know if that's true or not. That's what I'm wondering, really, if that is what's happening.
You know, Chris's experiment is very interesting, and what it makes me think of is, you know, we treat these spaces as completely separate, right? So certainly I was thinking, okay, there's transcriptional space, which can say which thing you can metabolize, and then there's motion and three-dimensional space. But maybe that way of cutting up the world is just not right in that case, right? Maybe both of those are parts of the same space, and we're just, you know, we're seeing them as two different spaces because whatever, that's how we view, you know, that's how we've cut it up. But maybe because for the bacterium, those are actually a kind of invariant. They both solve the same problem. Maybe they are part of the same space, and it's not objectively valid for us to say that these are completely different spaces.
Yeah, if you think of the bacterium as its only spaces perhaps are concentration spaces for things that it likes or doesn't like. Maybe those are the only dimensions it's really interested in.
Yeah, in that case, movement and transcriptional change are just parts of the same set of, you know, the same superset of actions that it can take to manage what it cares about.
There must be someone that did this experiment, just not on this whole framework that we're right now discussing, but there's got to be someone that must have looked at, were the organisms that moved around, and did they come with transcriptional changes whereas the other ones, and try to separate a population? I mean, it might just be that's the origin of like one origin of, you know, diversification and evolution, that you just basically had organisms that where the mechanism was implemented more strongly in one space whereas the other, and that's how they ended up in like a Darwin scheme, creating their own niches essentially. Not that doesn't mean that they were actually separated fundamentally, but they were basically, again, separated more in the timing and the efficacy of those different strategies, and then so in the end one was more emergent as a dominant space essentially. But yeah.
Yeah, one might imagine that if you looked at truly wild populations of E. coli instead of at lab pet populations of E. coli, you'd find population variants that regulate things like chemotaxis differently. You might have populations that swim in a straight line longer, for example, before they stop and retest.
Mm-hmm.
Yeah, there is this example of minimally cognitive agents, basically recurrent neural networks, that were evolved to do two different tasks that have two different corresponding timescales. One is chemotaxis, the other is walking. So they have inherently different timescales associated with them, but it's actually the same neural network, all of the same neurons, artificial neurons, everything interconnected, but they evolved to have a separation of timescales. It's a dynamical systems concept, which the agent just learned to pick up, and it used that idea to match the dynamics in the same network to different tasks, depending on the different timescales and the context. So I think the idea of specialization exists in some sense, but maybe it's not as concrete as we might think, like, or as compartmentalized as we might think. It may be distributed and yet separated or specialized in a different sense.
Very interesting. Okay. Anybody else? Any questions?
I'm really itching to do a thought experiment, but maybe we don't have the time to.
Go ahead. Go for it.
Yeah, it's Robert. So it's a toy example, right? I'm thinking of, so you ran the toy example in a space where the norms are invariant, right? So that's sort of your premise. And imagining what would it look like if you ran that agent in a space that can be distorted, like example near a black hole or something. So depending on the objects in the space may be distorted, and your norms are no longer invariant. Now, your agent itself exists in that space, but perhaps the question is it possible for that agent to learn or infer the rules of distortion? So it goes back to the relativity principle, right? There are a certain set of rules, Einstein's theory, that says space can be distorted in certain ways. So it's almost like meta inference. Do you think an agent that lives on physical space can possibly infer this meta rules that dictate how the space itself distorts? Do you think such meta inference is possible in your toy model?
Well, I think, I mean, at least locally, I mean, if you talk about this special theory of relativity stuff, I mean, there are these global curvature ideas, and it's probably very hard to track with such a model. But at least locally, yes, I don't see more. Maybe one has to try.
So would it require some sort of special sensors to do that, or like meta sensors or something, or is it just a second order? So right now your agent tracks first order differences. So
yes, exactly. I mean, right. I mean, it's a bit. It probably has to do with this very simple criteria which are used in that particular example. That ds is 0 in some sense, and then you can calculate. Well, 0 is just a regular norm which you can calculate, and you can replace that with something more sophisticated. Of course, the question is, if you talk about more interesting spaces, you would like to want to relax that and think about, well, how could one make an example where one is not fixed on one particular rule of finding these invariances, but can, for example, just use a general function of multiple order derivatives or whatever. And it's interesting. I don't know to what extent one could make that work. I mean, what one of course could do, one could try out different things and see whether one of the criteria or whether one member of this set of criteria works, and then kind of say, oh, okay, what works kind of reflects that particular weird structure of this underlying manifold. But I wonder whether there is a more principled way of approaching this problem. And I must say, I don't know. So I'm pretty sure that it works with this somehow. It's probably kind of involved to go through these different kinds of criteria, but it would work in principle. But a nicer, more principled way to find that would be desirable, I guess.
I think it's kind of related to what we have been discussing, right? The agent lives in physical space, but there is this different kind of space that exists, and that's where all these rules of spatial distortion comes into play. And for an agent to be able to detect that space or even be aware of that space I think that could be another toy experiment that you could try that may be related to all the discussions that you have been having on the relationship between different spaces. This is just another space. So maybe it's something simple to set it up. I don't know.
I mean, what I actually tried in my toy model is to kind of relax or use different conditions for finding these invariants. But I didn't really do it systematically or find something systematically. But it would be an interesting direction to actually look at. It might connect to this idea.
Great. Anybody else? Okay, cool. Well, thank you so much. That was really great. Lots of good discussion.
I thank you very much for having me here. It was a great pleasure to be here.
Yeah,
I hope we can discuss soon again.
definitely. Okay,
Okay.
thanks everyone. We'll see you
Thanks,
next time.
Robert.
Oh,
Bye-bye.
thanks.
Bye.